everybody should take. So this is the course link. Let me post the link again in the chat box. It's called the Introduction to Bioinformatics course. Once you come across to this course, you will notice that this course has an introductory video. So this is if you click on this first link, you will notice that it will take you to the first lesson of this course. So this lesson starts with a form. You may have already taken the Introduction to Bioinformatics course, or you may just be starting out. This form helps us to understand more about who you are and what your interest in relation to bioinformatics, biology, or biotechnology is, or any other related domain. So we have also your research interest that you can fill out right here. So please go ahead and fill out the form, and then you would be, once you submit it, you would be able to start with the course. And you will notice that there are a lot of images, there's a lot of text that defines what is bioinformatics, what are the different applications of bioinformatics, and each section you have to complete to move on to the next section. So, right, so this is the Introduction to Bioinformatics course that I hope that everybody here would be able to go ahead and look at. And now for everybody who is also interested in RNA-Seq. So this is the Transcriptomics courses. Now let me also post the link to this course in the chat box. Here you would find that the first lesson is the biology of transcription. And then we have RNA quantification, RNA-Seq and NGS sequencing. And each of these lessons start with a video. So you will be noticing that there is a video here that you can first play to understand what this course is all about. And I also want to introduce you to our Slack channel. So this channel is for our participants only. What we do as part of the Slack channel is help participants communicate with our team in an effective manner beyond the sessions. So these are the few resources at the beginning that we will be using. And now let me go back to my presentation and take you through to the next resource. So one of the other resources that we will be using is the TBioInfo platform for bioinformatics processing and analysis of RNA-Seq data. The platform will include demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections of this multi-omics platform. And we will also have exercises designed to explain code for these types of visualizations in both R and Python, from the very basics to the cutting edge loading, transformation, visualization, as well as complex discovery of patterns in real data sets used by researchers to study and understand biological phenomena at the molecular level. So once our understanding of RNA-Seq is clear from the courses, exercises, and lessons, we will also use curated data sets on malaria to study the efficacy of vaccines and drugs associated with resistance and the complexity of host pathogen vector interaction. Our team has already prepared a number of research data repositories and databases that will help participants identify high impact data sets for analysis. Besides malaria, we will discuss various applications of RNA-Seq to the study of SARS-CoV-2, where selective pressure on various proteins will lead to the emergence of new variants. Such changes are already being seen as a result of increased immunity, vaccination, and other host pathogen dynamics. This whole process will help you to apply these methods that you will learn to curated and new data sets and develop independent projects that showcase what you have learned in the context of a personal portfolio of projects. As a result, anyone who is starting on this journey from today can start developing a project of their own, like our previous OmicsLogic participants, all of whose projects are displayed here. And this is exactly how we will be using a combination of online sessions, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective in our OmicsLogic programs. So starting from today, our participants will be interacting with our mentors. And if you are new to a platform, let me introduce you to our mentors here. We have Dr. Harpreet Kaur, who is our OmicsLogic trainer and an expert bioinformatician. She mentors the sessions and will guide you through the program. Her area of specialization is in genomics. Dr. Mohit Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. He will also work with participants on their research projects. Ilya Brodsky is our CEO and co-founder. His experience in research and academia has led to many research labs and university collaborations and has been recognized for the simplified approach to omics analysis for education. Now, I would like to invite Ilya to speak more about transcriptomics for infectious diseases. Over to you, Ilya. Thank you.
Thank you very much, Bipsa, and welcome everyone to this first session. Before we begin, let me just briefly introduce myself. My name is Ilya Brodsky. I'm the co-founder and CEO of Pine Biotech. Our vision is to enhance human health by enabling biological research and discovery with relevant data, solutions, and expert support. And so this program is really designed around simplifying some of the bioinformatics complexities to allow as many participants as possible to gain a better sense of how to apply bioinformatics tools to specifically RNA-Seq transcriptomic data in the context of infectious diseases. So as a company, our mission is to simplify bioinformatics and advance research through our modular and intuitive multi-omics platform that is powered by human experience and artificial intelligence. And so this platform was developed at the Tauber Bioinformatics Research Center, which is focused on development of novel algorithms, as well as making them user-friendly so that anyone like you or I could actually try and use and compare and decide which algorithms are actually needed for the analysis that we would like to do. But what we've discovered is that this actually does require a serious approach to how we present the different challenges, research questions, and these tools in the context of the logic of analysis. And so that's why this program is called OmicsLogic. And specifically, the program that we are starting today is called OmicsLogic Transcriptomics. So what I will do in my part of the presentation is expand a little bit on some of the topics that we will be going through to set the stage for this training program. So the training will be focused on transcriptomics, specifically how we can leverage RNA-Seq data to study infectious disease from the perspective of host response, disease progression, and host pathogen vector interaction. So to do this, we will discuss the various aspects of RNA-Seq data analysis, including how to convert raw reads from sequencers to structured data, how to perform exploratory analysis and clustering to understand some patterns and what we are looking at in the data as a whole, how to identify important genes through differential gene expression and feature selection, how to understand biological signals through pathway enrichment, and how to perform single-cell deconvolution of bulk RNA-Seq and analysis of single-cell RNA-Seq data. So we really have a lot planned for this program. So as a result of completing this program, we hope that each participant will gain a clear picture of RNA-Seq data processing, analysis, and biological interpretation using big data analysis cloud-based tools. We will also learn about how machine learning is used to find complex patterns in big data and how statistics could be applied to such data to extract meaningful insights. What's important is that this program is best suited for those interested in research projects. So you will have the opportunity to develop an independent research project question or proposal. After completing all of the program sessions, you will earn a certificate of excellence if you have designed and completed an advanced project or a certificate of participation for completing the basic training. And so today is the first session. And what we will do in today's session is just start talking about what is RNA-Seq data and what is transcriptomics, and how do we actually get started to think about the measurements that we are going to be taking with RNA-Seq data. Now, for those of you raising your hand or trying to ask a question, I will pause for questions after a little bit of an introduction. Okay, so let's start with some basics. What is transcriptomics and how is it different from other omics? Omics technologies, like next-generation sequencing, can be used to explore the roles, relationships, and actions of the various types of molecules that make up the cells of an organism. So there are many types of omics data that could be generated using a variety of techniques. One of them is next-generation sequencing, and it is also known as high-throughput sequencing. The data from high-throughput sequencing can reveal important information about genomic variants in genomics, epigenetic regulation mechanisms like epigenomics, but transcriptomics is needed to generate data about mRNA expression, and it allows us to study patterns or levels of gene and isoform expression, including how these genes define pathways or how they are modulated through different levels of gene expression or through alternative splicing. mRNA expression can be studied using a variety of methods, and this includes northern blot analysis, ribonuclease protection assay, and quantitative reverse transcriptase coupled polymerase chain reaction, or RT-PCR. So what are these methods, right? Before we kind of jump into next generation sequencing. So northern blotting is a very relatively easy method to use and has become one of the widely used methods for characterizing mRNA size and relative abundance. Northern blotting is the only one of these techniques that allows mRNA size determination, and so it has been a method for detecting mutations that result in abnormal mRNA size. But as a quantitative technique, northern blotting represents several limitations because of its inability to control for the efficiency of RNA transfer and membrane binding. And so it is a semi-quantitative technique that is most suitable for determining relative concentrations of mRNA species that occur in moderate to high abundance. So then ribonuclease protection assay, RPA, is right here. So RPA is performed by hybridization in solution of labeled RNA probe to an RNA sample containing the target RNA. And after hybridization, excess single-stranded probe and non-target RNA is digested by treatment with RNase mixture. The protected probe target RNA hybrids may be analyzed by denaturing polyacrylamide gel electrophoresis, PAGE, or by precipitation and filter binding for quantitation by scintillation counting. Okay, so structure of a typical transcription template for synthesizing an RNA probe. cDNA sequence corresponding to the target mRNA is subcloned downstream of a bacteriophage RNA polymerase promoter. So one of the aspects of this method makes it a semi-easy method of using this technique to quantify mRNA, but it is prone to mistakes and various technical issues. Another technique, like I mentioned, was RT-PCR, which is a technique of choice to quantify low abundance mRNA species and detect mRNA in small number of cells, because as you can imagine, PCR amplifies the low quantity RNA, so it allows us to detect what is in there. And so it should detect cDNA derived from a single mRNA molecule. And so in practice, even though it should detect even one, 10 or more mRNA copies are required because of the relative inefficiency of the reverse transcriptase reaction required to convert mRNA to cDNA for subsequent amplification. And so what all of these techniques led to is the emergence of what's called microarray. And microarrays are also hybridization based. And so what that means is that microarray hybridization assays are widely used for a variety of research and clinical applications where a known mRNA transcript is needed. Unlike most historical platforms for clinical RNA measurement, these microarrays can contain thousands and thousands of predefined mRNA probes. So even specific splicing transcripts could be included on these probes. So how is RNA-seq different from all of these methods? RNA-seq, as opposed to all of these kinds of platforms that we already discussed, RNA-seq has a wider dynamic range. As you can see right here, what's important about RNA is when it is translated, there's an alternative combination of exons that are spliced together to actually form the transcript that will become a protein through translation. And so this is called a wider dynamic range, which means that you can detect many more types of variations, because as opposed to a textbook understanding of this alternative splicing, actually it can happen in aberrant ways, right? In many different ways. In cancer, for example, sometimes introns are known to be included in the actual transcript. And so you would have no way of detecting that using these previous first generation techniques. So RNA-seq is really a powerful method that avoids some of the technical limitations, such as varying probe performance and cross-hybridization. RNA-seq can measure the expression levels of thousands of genes simultaneously and provide insights into functional pathways and the regulatory networks in biological systems. In addition, RNA-seq can provide novel insights into alternative splicing, unannotated exons, and novel transcripts. And most importantly, such detail has already proven to yield significant discoveries in many research areas, like I mentioned, in cancer and others. So actually, in our next session, we will cover a lot more in detail the actual process of next generation sequencing, how this data is prepared, but I'll briefly touch on it today as well. So next generation sequencing technology captures DNA sequence and generates digital data, right? So the objective here is to take a biological sample, extract the mRNA, convert it to cDNA, and then use pieces of sheared cDNA to prepare a flow cell that is full of these prepared sequences that could then be amplified through a solid phase PCR bridge amplification. And then from reading of the signal by converting each letter to a specific colored signal, we can get accurate reading of the signal coming from each cDNA. So the machine uses image analysis to capture each letter in the flow cell fragments by analyzing visual patterns and converting them to a sequence of letters. Typically, next generation sequencing, okay, I think my screen got turned off. Okay, yeah.
We can see your screen here.
Yeah, okay. So typically, the reads coming out of the sequencer are anywhere between 30 and 300 base pairs long, even though today, with the evolving technology for RNA sequencing, very high lengths of reads can also be achieved, thousands of nucleotides per read, and that's an exciting new development in RNA-seq. Importantly, RNA sequencing allows for the detection of a wide variety of RNA species, including mRNA, non-coding RNA, pathogen RNA, chimeric gene fusions, transcript isoforms, and splice variants, and provides the capability to quantify known predefined RNA species, as well as discover novel and rare RNA transcript variants within a single sample. In addition to differential expression and detection of novel transcripts, RNA-seq also supports the detection of mutations and germline variation for hundreds to thousands of expressed genetic variants, facilitating assessment of allele-specific expression of these variants. And so here, we will focus on the application of RNA-seq for infectious disease diagnosis. For example, in RNA-seq samples, you can extract enough reads to even detect presence of a particular pathogen. For example, RNA-seq is used to detect clinically important RNA viruses in regular screening of tissues like HIV, Ebola, West Nile, dengue, hepatitis A, B, and D, Coxsackie and influenza viruses, as well as severe acute respiratory syndrome, SARS, and Middle East respiratory syndrome, MERS, coronaviruses. A lot of these applications are typically not the typical approach to study RNA-seq, but we will discuss how RNA-seq could be used in that. So specifically for pathogen mRNA measurement, it could be useful as a marker of infection, right? So you can detect, for example, low abundance of viral DNA or RNA present. And it can be specifically important in cases of low-level infection, right? So for example, bacteremia and cerebrospinal fluid infections are very important for low detection. And it could be a better predictor of disease compared to direct genomic detection. For example, the simple detection of human papillomavirus or HPV DNA is not sufficient to diagnose HPV-related squamous cell carcinoma. Even though HPV DNA is detectable in about 14% of healthy control women. So RNA-based diagnostics to detect HPV have been developed. So there's a lot of important applications and considerations to not just detect a particular DNA sequence, but actually to look at the expression of specific genes of these pathogens. On the host side, we can also look at the host response. For example, mRNA signatures that could be useful to monitor even specific infections. For example, upregulation of specific host immune factors like interferon beta-1 or IFNB1, interferon lambda-2, IFNL2, and interferon lambda-3, IFNL3, were recently demonstrated for genotype 3 HCV infection, which is associated with accelerated liver fibrosis and is an independent risk factor for hepatocellular carcinoma compared to non-genotype 3 HCV infection. Host microRNA gene interactions during the infection response are also proving to be a fruitful source of potential diagnostic biomarkers for specific infection, and even distinguishing between active and latent infections, as we know in hepatitis. For example, the use of such biomarkers to discriminate between latent infection and active disease with M. tuberculosis. Right, so there's a lot of applications of RNA-seq for detection, accurate characterization of pathogen-caused diseases, as well as measurement of host responses. We know in COVID, for example, we can distinguish between acute disease stages as opposed to mild disease stages using RNA-seq data as well. And so obviously, in many of these, the challenge is to integrate and explore detailed data using a cloud platform that leverages big data analysis on the cloud, as opposed to more visualization and statistical analysis of your data on the laptop. And so Beeps already mentioned this platform. When you are registering on the OmicsLogic website, when you go and you make your registration, you should receive an email that will also come with password details. So when you go to log in, if you have not done this yet, and you create an account right here, once you sign up and fill out these details, and please follow up with an email check after you do this, you will actually see your information to log in to the server platform. So to kind of understand some of the specific details that we will cover in the program related to transcriptomic data analysis, please go with me to this link and let me know once you've had the chance to log in. Okay, so this is the link. And please put in number 1 or yes, or something like this in the chat box so that I know that you are with me here on this platform. Okay, I'm just going to give it a couple of minutes. Again, if you don't have access to the platform, you don't know what your password is or how to log in, just simply registering for free on the learnomixlogic.com platform will allow you to get that in your email. Okay, thank you, Rashid. Rashid is already here. Has anyone else been able to? Okay, thank you, Dua and Timotopy. Okay, thank you very much. Let's wait for at least a couple more. Okay, excellent. Okay, so as you can see here, we have the areas of analysis. This is the section for processing next generation sequencing data. And here, the first one that you will see is transcriptomics. And you can hover over some of these icons to kind of learn more about each section. So let's first look at this RNA-Seq chip seq. And so here you will see a few different examples that we will look at today, just to kind of understand what we will learn in the full course. Typically, when we do our training in transcriptomics, we go through this first example with cell line projects. And so this cell line project is a cancer project, but today we will speak about applications of transcriptomics to infectious diseases. And so let's look over here at this demo called SF9 Unmapped Reads. Okay, so once you click here, you will get into a demo section of the platform. In other words, you don't have to upload data. We'll introduce you to some data sets in just a little bit, but for now, you can just go to this section. Again, for those of you that are following along, right here, it's called Demo SF9 Unmapped Reads. Okay, so once you are here, you will see that each button here corresponds with a step. And so each step allows us to identify, okay, actually, sorry, I see that there's something, let's go to another one. Let's go to this one, cell line. Yeah, I see that there's some issue with that demo because I didn't see anything pop up. So, okay, so here, again, we will see an overview of some of the important logical steps for RNA-Seq data processing. And you can see that there are actually several options here that will open up once you create this pipeline. Okay, so here we have start, and I can choose to go to any one of these sections. Now, a lot of times when you think about processing RNA-Seq data, you're thinking about mapping the host data onto the reference genome to understand the alignment as the first step. Yes, Rashmi, did you have a question? Okay, if you do have a question, feel free to put it in the chat box. Yes, go ahead. Okay. So one of the things that I want to point out is that typically before alignment itself, it is useful to pre-process the data to clean it from certain artifacts. We'll learn in our next session why we do that. So let's go ahead and clean up right here some of the adapters and that PCR amplification from that bridge amplification. And then we will see that we have two options for our next step. One is HiSat2, so let's select that one. What HiSat2 does is it improves on the previous algorithms called Top Hat and Top Hat 2, and allows us to do an accurate alignment with alternative splicing junctions. Excuse me. And what you will see here is that one important output from this HiSat2 algorithm are some unmapped reads that have already been cleaned up for each one of our samples. So whenever we're looking for a pathogen, potentially that has some transcription already for one of its genes, we want to identify that by looking at reads that have not been mapped to the reference genome. So if this is a human sample, we will take the human sample and try to detect unmapped reads that can lead to discovery of those pathogen reads that we can then detect using mapping to a database. And so we'll talk about that in one of our future sessions. But here we'll go to HiSat2. You can see that what it combines is mapping on genome and mapping on junction to detect exons. Then we will construct isoforms by clicking here, which means that we will detect how many different transcripts are available in our data by linking together all of the present exons using those junctions of splicing points on the genome. Then we need to create a GTF file. We'll talk a lot more about the various types of annotation steps that are important in this pipeline. And typically what we think about of a GTF file is an annotation of all of the important positions that we look for when we analyze our RNA-seq data. So we'll go through this step for GTF processing. And now that we have this GTF, we can actually map our reads for quantification. So here we are going to perform mapping on transcripts using bowtie2t and create a table of expression using RSEM, or RNA-seq using expectation maximization. So this is a a method to understand significance of amplification and do some downstream quantification with normalization for our gene expression table. And so after we do this, we can perform differential gene expression, and this differential gene expression will help us detect which genes are differentially expressed in our data using DSeq2 or EDGE-R, and you will see that some of these outputs are going to be very easy to read. And so the next step after this, what we recommend doing after today's session, for those of you that have completed the registration, is to start learning about each one of these specific places that we are going to be doing throughout this program. Okay, so here you will click on end, and now you can see some of these outputs from DSeq2. You can see that there are some mapping statistics, DSeq for expression of genes, isoforms, and you will see all of your results from RSEM here as well. So this is an important part because the input files here are actually gigabytes and gigabytes of data that would not be practical to use on your local computer, on your local system. Okay, so does anyone have any questions about what we did so far? I see a question, will this recording be made available to us after the session today? Yes. Beepsa, if you could please take the link from YouTube. We have this streaming live right now, so you will be able to see the recording after we are done. Maybe Beepsa, you could post that there as well. And we will follow up by email to those of you that are interested. Okay, any other questions before I move on? Okay, so what this platform will allow us to do is to start applying some of these RNA-seq methods to data sets that we might be interested in exploring. Now, what types of data sets are going to be curated for this program using various infectious diseases that we're going to be studying? One obvious interest that many have is the understanding of SARS-CoV-2 infection, disease progression, and variable clinical manifestations of this disease in patients. As you can imagine, any virus is a complex protein machinery that actually uses in a very effective way all of the encoded proteins to make a variety of tools to subvert how it is recognized by the immune system, what responses it causes in a variety of tissues, cells, and organs, and how it actually hijacks the machinery of the cell to produce its own virions. And so we'll take a look at how RNA-seq data can be helpful in analyzing such gene expression profiles from infected cells and cell cultures to understand the complexity of this protein machinery. We will also look at how SARS-CoV-2 interferes with the typical biological pathways of this immune response to evade the immune detection and to modulate the immune responses to achieve its goal of infecting cells at a rapid pace. We'll also look at how the design of experiments could be used to modulate different genomic construction of viral genomes, and then study the application of the response in the host so that we understand what is the function of particular proteins and genes encoded in these viral genomes. Importantly, we will look at some of the standard tools like we did today. We'll talk about how to use statistical analysis conceptually to start studying the different variable infection and specific types of conditions that were designed as a part of the experiment, all the way to more advanced and complex methods like regression that could be used to detect a variety of outcomes, including factors that influence our observed data and the outcomes that we can observe through integrating with clinical data or our biological interpretation of that data. And so we will use gene set and pathway enrichment analysis to then talk about biological interpretation of what we observe in these conditions that we are studying. There's a question. Okay, so yes, if you're just joining, and that's a good place to kind of talk about. So we are talking about this transcriptomics program. It's starting today. This is the first session for this training program. What we are doing in this program, first of all, is I have briefly described what the program will cover and what we will be talking about, but this program is actually going to be quite comprehensive. So we'll take some more time for us to go through all of these topics. And the topics that we will cover is the NGS RNA-Seq technology and applications, how to process raw data and get a table of expression, how to perform differential gene expression using statistical considerations about multiple testing, how to identify statistical correlation and regression, use data science for omics using R and Python, which we will cover at the later part of the program. And then also how to identify data sets to start practicing what you learn and actually develop a project, a research project of your own. So hopefully that answers your question there as well. So for the purposes of time, I'm going to skip over some of the additional methods, and I want to pass it on to Dr. Harpreet Kaur, who is going to be one of your program mentors, to talk about some of the curated data sets that we have prepared to allow us to apply these RNA-Seq analysis methods to infectious disease projects. So Dr. Kaur, I'll pass it on to you now.
Thank you, Ilya. And can you make me just co-host? Okay, thank you. Hello, everyone. So today we are going to talk about what are important data sets are there regarding infectious disease, especially related to the transcriptomics data. So here we have like the first repository that I'm going to talk about. We have already curated some of the important data sets and that I will share. So the first repository that we are going to talk about is Geo. So here you will find a data set. So there are some of the important things that I just want to mention. So here under the series matrix file, just give me a second. Just give me a second. Okay, so let me first explain you actually on Geo what kind of information is there and how you should proceed to retrieve a data set. So if you already have some Geo ID, which is also known start with GSC. The GSC means series ID. So you can search here under the Geo session number. So then here you will find like actually this data is related with which study. So in the title you will find that, and then they also describe the summary. So like from which study this particular data has been taken, and actually what originally authors have done with this data. So then under the overall design, you will get information regarding kind of how many samples are there. For example, here you can see like with this specific data set, here are 108 samples that are paired pre and post challenge for 54 individuals. Then 198 data sets are again for the pre and post challenge for 99 individuals. So they are it contains two types of samples. Then under the citation, you will find the complete link of the original publication from which this data has been taken and submitted to the GEO. So then most important thing, actually where you will find this data, important where the metadata is available and where there is raw or processed data. So the first, the already original raw data, under the relation you will find this link like bioproject and SRA. So this is especially if you are interested in high throughput sequencing data. So the raw data is available either under the bioproject or SRA. So you will find those links related to your high throughput raw data. Then is about metadata. So here the third file under the download family, which is series matrix file. So here you will find metadata regarding high throughput or microarray data. So specifically if it is microarray data, in that case you will find the process data as well under the series matrix. So then here under the supplementary file, you will find the process data for high throughput sequencing while raw data for any microarray study. So then now I think let me share some of the data sets with you and also demonstrate to you how we can actually obtain the data set. So here I have provided some of the links and let's directly go to the repository. So just let me know if you're able to log into the GEO. So first, let me show you how actually we can find data from the GEO, which is Gene Expression Omnibus. So let me know once you are here. So type yes if you are able to go to GEO, or which is Gene Expression Omnibus. So let me paste its link in the chat. Okay. Okay. So here, for example, if you are interested in, malaria or some infectious disease like COVID-19, so first you need to create your query. For example, here I am searching for a malaria. So I just created my query directly malaria. So once I created that, and I will get this link. So the first I need to click on this number, like nearly that is 4046. So if I click on this number, so I will direct it to this interface on the GEO. So then from here itself, I can select top organism. So on the right side of this interface, you will, if you are interested like in a particular kind of organism, you can select that. For example, if you want to extract only those data sets which are associated with the Plasmodium falciparum, so you can select this. So then what kind of data that you are interested, whether you are interested in high throughput sequencing or microarray data. So what we can do under the study type, if you just click on the customize, and this pop-up will come. So here you can select whether you are interested in high throughput sequencing data or a microarray data. So under the expression profiling by array is data generated by microarray profiling technique. And like expression profiling by high throughput sequencing, one can find data here. So then if you're interested in coding or the non-coding, you can select those. So then you can go to show, and then you need to click that particular your selective filter. For example, I had already selected expression profiling by high throughput sequencing, right? So here you will get like, now we have data set, nearly 39 data sets that are associated with the expression profiling by high throughput sequencing, specifically related with the Plasmodium falciparum. Now, as the number is reduced, like now we are left with only 39 data sets. So one can explore these kind of data sets individually. So for example, the first data set is associated with the single cell label. So one can, if somebody is interested in single cell, similarly here. The second study is also again associated with the single cell sequencing, transcriptional heterogeneity and stochastic gene expression in Plasmodium schizoides. So someone, if you are depending upon your research interest, you can take these data sets. So I had already find few of some of the important data sets here. For example, this particular study here, let me share it link with you. So I have already curated like nearly 10 data sets related to the malaria. So let me paste link for one of the data sets that to me it looks interesting. So here, if you look at this particular study, here you will get details regarding the data set, like this data is associated with which study. So here the molecular hallmarks of naturally acquired immunity to the malaria. So then you will find what authors have actually done under the summary. So they have tried to understand the immunity to malaria can be acquired through natural exposure to Plasmodium falciparum, but only after the years of repeated infection. Then typically this immunity is acquired by adolescents and confers protection against disease, but not infections. So efforts to understand the mechanism of this immunity are integral to the development of a vaccine that would mimic the induction of adult immunity in the children. So they have like, then you can go to details, like here under the citation, you will find complete publication. So here, let me share it link as well. So this is like whenever you actually going through a data or taking a data, you need to go through original publication. What authors have already done with this data, so that when you actually asking your question, you should not be like asking the same question which already authors have addressed, or anyone who have already addressed some maybe in a different study. So you need to make sure that your research project should be noble enough. So now let's come back to the data set. Here under the overall design, you will find how many samples are there. Then here the data set that I was talking about, like here, we have FPKM data. So under the, if I already look at what is this data. So here we have total 198 samples. So if you click and you can find all the samples. Just to understand actually how this data is processed, if you just click on one sample, you will get complete information regarding how this data is processed. For example, here under the data processing, authors have explained, so what they have generated, they have FPKM values were obtained from the Toffert algorithm. So then they also use a genome build, which is GRCH37. So then also they explain which version of Toffert they have used. If you're looking at your sequence reads are mapped, so using the Toffert version 2. So you will get this information. Then the supplementary data, what supplementary data contains. So here, because for the high throughput sequencing or techniques, on the geo under the supplementary data, you will find process data. So under the supplementary file, but for what it contains, so it is a tab delimited file, include FPKM values for the gene and transcript isoform, and read count per gene. Then what kind of information they have, the metadata information for this particular data. So here they have under the characteristics, you will find that information. So here, if you look at the characteristics, they have provided information regarding two important characteristics. One is time point, that is post-infection. Second is infectious agent, like here, that is Plasmodium falciparum. So this is regarding the one sample, if you just want to understand what is this data. Now go back to the complete data. So here under the series matrix file, you will find metadata. Now let me download this data. So you can download and find the metadata here. Then the supplementary file is here. So similarly, if you are interested in raw data, so you will find links for the bioproject. Now let me take you and show you these bioprojects and SRA. This is SRA, where we will get sequence read data. So this is the bioproject for which you will get link for both for SRA and what is this project is about. So you will get complete details here as well. So then this is SRA, that is sequence read archive. So you will have the FASTQ file data here, right? So now let me show you what kind of data and how we can actually generate a simple matrix from the series matrix and what kind of metadata is there. So let me take one example. Just give me a second. Okay, so here this is the series matrix data, right? So the series matrix data that which contain metadata. So I have pasted that in an Excel because that is ggzip file. So while unzipping, we will get this metadata. So here you will get first the top part contains information regarding the study itself. Then here, then it starts with sample, like here, sample title. So from here onward, you will have information regarding each of these samples. So for example, here, so this is like another data. So here we have like sample title, then under the sample underscore characteristics, which contain actually the metadata information. So this is like if I just copy these sample characteristics and this ID. So I have already copied this here. So now I have important metadata regarding this particular data set. So let me share its ID as well. Sorry. So here you will get like time point, right? Exactly the day, outcome. So all the metadata information is available in the series matrix. So you just need to, like here again, against the sample ID, here in the geo, they usually give always a sample ID with the GSM. So the sample ID always start with the GSM followed by some number, which is always a unique number. So then you will find these are different clinical or demographic characteristics related to the specific data. So then this is the data that we have. Like here, they have provided the gene symbol in the first column, followed by this entries ID and gene ID. So from here onward, if I'm looking at this particular data, here we have these samples. So in the columns, we have samples except these 4 starting rows. So these sometimes only authors provide like only transcript ID, or sometimes they provide additional information like gene name, that is gene symbol, rather than not a name. So then gene ID. So this kind of information, depending upon authors, sometimes provide you this information as well. So here onward, we have the sample IDs. So you can always match these sample IDs with the metadata. For example, I am looking at this particular ID. Where is this ID is available? So what I can do, I can look at my metadata, which I have prepared. So this is this sample. Similarly, we can actually map our all the IDs with the metadata information. Then we can ask the different questions depending upon what information we have, right? So similarly, I have like already curated like nearly 10 data sets. So similarly, you can find another data here. This is this one. So this is regarding transcription response to RTS standard regime and this particular treatment vaccine in controlled human malaria infection. So similarly, if you look at this data, here we have this information like this data is taken from 46 individuals, and they have like already included nearly 670 samples. So each of these samples, this is like very large data. So if you like somebody is interested in supervised machine learning, in that scenario, if you want to prepare and want to generate a good model in that, in such scenario, usually we need large number of samples. So this kind of data is very useful when you are doing that supervised machine learning. So then like here, I have already shown you under the supplementary file, you will get the process data. So particularly in this study, so let me share this data as well. So this is again a high throughput sequencing data. So where you can look at like this is a high throughput sequencing data, expression profiling by high throughput sequencing. So then here under the supplementary file, you have two types of data. One is log2 expression values. So other one raw count value. So this is already TXT file and they have produced the tables. So then if you want to look at what are the important metadata information you will have. So you can again, just to check before downloading anything, you again check under the characteristics, you will find this information like patient ID, gender, ethnicity, whether it's African, heritage or African American, then the what is treatment is given, then whether the protection is already there or not. So they have provide this kind of information. Right. So then you want to look at how this particular data is processed. So authors have used align star for the alignment. They have used the star algorithm. Then sequence reads was filtered for low quality sequence in the presence of N, then mapped to the HG19 whole genome using star aligner, particular this version. Then they used gene counts were calculated using HT6. Then how the data is quantified, as Ilya has already explained you, like there are the number of different steps for the pre-processing, which removes duplicates and adapter sequences or any non-technical sequences. Then your clean reads are mapped against and reference genome or reference transcriptome. Then you quantify the gene expression. So what are the algorithm used for mapping or quantification? They have explained that. Then in the supplementary data, they have already normalized gene count tables. So this kind of information you will get, and then you can actually analyze such data, right? So if you are interested in raw data, you can always go to the raw repositories. But the challenge with the raw data, for example, here I was showing you, even just while looking at single file. So the single file is itself like this is 3.4 GB, 4 GB, this is more than 4 GB. So like this is 3.4, 2.8 GB. So even a single file is difficult to handle. So to process such data, we have our server, which will simply generate the gene expression table, which Ilya already shown you. So here you need not to download your FASTQ files on your system. So we just need to create a simple SRA run table. So how I can generate SRA run table? Simply I need to go to, so this is my already data here. So what I can do, so this is the data if you want to use for the public repository. Here you can use run info, then you can create this file.
Hello. Oh, बाप रे, क्या हो गया है?
Please go ahead, Dr. Harpreet.
Okay, so here we have, like after creating the SRA run table, we are not downloading complete or any of the FASTQ files. What we can do, we can generate our simply SRA run table. So let me just give me a second. So here I have already generated my SRA run table. So what I can do, I can give just this SRA run table to produce simple gene expression table. So for example, after so this is my SRA run table. So which I just need to copy in some txt file. Then I can actually give this table and process this such a high, like here we have 100 samples. So even on an average, if I just consider that one file is like 4 GB. So you will have, otherwise you need to download like more than 400 GB space, even to keep this data. So to tackle such things, what we did, like you just need to provide this table. And if I just here with the, again, just with the demo, I can show you like this in this, sorry. Here under the NGS data, particularly you will find transcriptomics subsection. So here these drop down indicates the demo pipeline. So for example, here we have already uploaded one data set that is related to the another oncology project, that is PDX project. So if I just go to this demo, we have already uploaded our data, input data there. So what we can do, simply start. By start, I am able to upload this data, which is already loaded because this is demo pipeline. Then is pre-processing. So here the Tremomatic, the first algorithm, that is this option getting highlighted, what it's doing, that it will remove adapters from your raw sequencing reads, right? So then after removing the adapters, then we will go to the PCR clean, which will remove any duplicates reads here. Then after the clean reads, next is how you are going to align your reads against the reference transcriptome. So here number of the algorithms get highlighted, like Botai 2G, then HiSet, similarly there is Stockford, then this is Botai 2T. So to align my data on reference transcriptome, here I am using Botai 2T, which will align my reads against reference transcriptome. So then followed by the gene expression quantification. So what I need to do, I can choose any of these algorithms depending upon the goal of my analysis, as well as what algorithm I have used for the mapping. So depending upon the mapping algorithm as well as our goal, we can actually then choose an algorithm to quantify that expression. So like here we can choose RSCM expression table. So then with the end, we will able to complete this pipeline. So here under the download in file, so here I have now I have already clear gene expression table, if you are processing raw data, right? So here we will get this kind of gene expression table. So now let me share this table would be similar to that we have obtained under the processing data. So this kind of table I have now. So this is FPKM gene expression values, and we will learn what are these FPKM values or what are these transcript IDs in our next session. So here my goal is how we can take raw or the process data from the public repository GEO related to the infectious disease, right? So let me paste it into the Excel itself. So, and let me show you. So here, if you look at this data, here we have ensemble IDs for the transcript, followed by in the columns we have samples, right? These are ER positive samples. So you have to look at what are those samples. So this is how we can actually generate a same gene expression table by processing. So if you have you don't have any process data from the public repository, in that case, you can generate gene expression table using this simple platform, Tibanchu server. So if you already have processed data, then you can ask these questions. What kind of question now I can ask? Like here I was showing you, we have a metadata. In the based on the metadata, now I can actually ask my question. For example, I just want to look at the difference between pre-vaccine and the post-vaccination changes, right? So this is like a vaccine is given to the patient. So then what are the transcriptional changes taking place after the vaccination? So we can ask this kind of question like pre or post-vaccination. So then we can also, for the post-vaccination, then we can look at they have given different days, different time points, right? So then we can ask questions, maybe already the people are actually vaccinated early. So what are the different transcriptional changes taking place in comparison to the subject which have given treatment later? So we can actually, depending upon what metadata we have, we can ask and create our research question. So that's, if anyone have any question, so let me know. And the participants who will join the program, then we can actually help you to create and design your research query based on the data set that we have curated. Like we have already curated maybe more than 10 to 20 data sets. Like here I was showing you for malaria. Similarly, we have a data set we have curated for COVID-19 and Ebola infections. So we can we will help you to design those research questions that you based on which you can design your own research project. So Vipsa, many people have the questions that how they can join research fellowship, and they message me about that. If you can explain them.
Sure. Yes. Sure, Dr. Harpreet. So, okay. So Suhanya has a question here. Could we also do the analysis with R using the same pipeline? If you can continue to reply to that question, I will also go ahead and share my screen.
Okay, so Vipsa, if you can share the screen from the code OmicsLogic, so that we can actually directly show.
Right. Let's go to learn them. Another course.
So like here under the Transcriptomics course itself. So if you just go to the Transcriptomics course.
Okay, give me a moment. I'll go to Transcriptomics.
So open that, and if you just find the lessons, click on the more.
Load more.
Yeah, load more. So here you will, like, we have various types of analysis: data visualization, PCA, differential gene expression analysis, supervised machine learning, and unsupervised machine learning algorithms. So we have also created the codes in R both as well as in Python. So you can also do, after you have a gene expression table, then you can actually run any kind of analysis, whether you are interested to produce, to generate a, or want to perform principal component analysis, or whether you are want to do differential gene expression analysis. But to process raw data, because it is not like, those codes are not available in R or the Python, because that is working at the in the behind. So there are different scripts. So those are actually different tools are working at the back end. So, but to further analyze such data, once we have a gene expression table, we have also generated both codes for R as well as in the Python. I hope it is clear to you now.
Thank you, Dr. Harpreet. So first, I would want to again take everybody through the curriculum once, and then get to some of the project questions that everybody has here, like how does one get started with their own project. So this is the syllabus. You will find it here again on this page. So the next session is on 19th, which is Monday. So we are going to be talking about analysis of transcriptomics data for immune profiling and drug response for response analysis for COVID-19 drug repurposing. So the COVID-19 and the malaria projects are example projects that we are going to be using to learn more about infectious disease research, but that does not mean that you on your own cannot do another project. So once we are getting started with the program and have progressed at least 1 to 2 weeks, what we will share with you is a project form where you can actually propose your own project. So if you are interested in working on a project, you can start by thinking already of what is the project title that you want to work on, or what is the project topic, and start looking at this literature that you have or you are interested in. Look at other research, as Dr. Harpreet was saying, that you need to look at the original publication to understand what those researchers have done in terms of this kind of data set, trying to answer this particular type of a biological question, right? So you have to start thinking. So if your end goal is to work on a project, you have to prepare yourself from now. What we are doing at this moment is first preparing you with the courses as well as these example projects in learning and understanding the logic, so that when you are clear about the logic, you're able to apply it to your own research question. So that is the goal. So we have started here on April 16th, and we'll meet again on Monday with our group of participants to discuss more about the COVID-19 drug repurposing. And then we will meet twice every week to talk more about transcriptomics data and walk through all of these different sessions. And then the program will end on May 20th. So you have almost over a month to work on not just the transcriptomics portion, but also work on your own project. So start thinking about it if you're interested. And if you want to join the program and you have not yet completed your registration, I wanted to quickly show everybody again. So this is the link. If you come, it's the same link. Just click on Get Started or the pricing option, and you will have these 3 different options right here. So again, I will explain what these 3 options mean. The beginner license will give you access to all of the resources, courses, lessons, as well as the project that we discussed today. The intermediate access gives you access to one-on-one mentor session with your mentor or whoever is walking you through this entire session. And the advanced license gives you an additional 30 days, which means that if you want to keep on working on your project, you can choose the advanced license that will give you access till June 20th. So then you are able to work on your project as well as go forward with it. So I see here is a question from MA Hashmi, can this help in studying cancer system biology? So we definitely do have programs that are dedicated to cancer biology. We just finished a precision oncology program, and we will be having another precision medicine program in the summer. And also next week, we have a program from AIMS, which is All India Institute of Medical Sciences. We are collaborating on a program on lymphoma. So we have those programs, but this particular program is dedicated to studying infectious diseases. So we are going to mainly be focusing on infectious diseases, but definitely all of these courses have a lot of data sets. So let me again go back to one of these courses just to show you some of these data sets and the lessons that are there. So if you notice, the first course here is the TCGA liver cancer project. So we have a lot of these courses that we have on cancer, as well as many of the transcriptomics courses itself has a lot of data sets on cancer. So definitely you can utilize these to study about cancer system biology as well. So as I introduced you to all the mentors, so you will know that Dr. Harpreet, Ilya, or Dr. Mohit will be mentoring the session. So it would be either one of them taking the sessions. And then mostly it would be Dr. Harpreet helping you out with the project and guiding you through the sessions with inputs by the other mentors from time to time. Okay, I think I've answered those questions. Now I wanted to show you how the research process looks like, and also give you like a little idea of what to expect from a project. So I'll just give you here some project examples. So we have a very well-defined process of doing or working on your own project that starts right with first learning from these courses that we already said, because they are going to give you the knowledge and the understanding or the foundation about what all these omics topics are, how they relate to each other. And then you start looking at your project. So here we have like a few guides on this page that you can understand on how to do a meaningful project presentation. So this is right for the end. But as we are going through the program, we'll also go through the different stages like literature review, then looking at these data sets, understanding what those data sets are saying as we are doing the analysis from those data sets. Then once this is done, what we have to do is present. So you will also notice that there are a lot of presentations, and many of these projects have been actually completed by high school students. For example, this project by Amelia, so she is a high school student. So anyone who is interested in working on a project can start with a program like this, where they first learn from a program which is of their research interest. So it could be metagenomics, it could be transcriptomics like this, it could be infectious diseases. And then as we move forward, you will be working on your own project where you work with your mentor, you find a data set, you start looking at these different literature. So in regards to that, you would be able to find out how to work through these. So we have these step-by-step processes to help you not only work on your project, not only propose your project, but refine that project so that it is presentable to a wider number of people. In all of our webinars and in also a lot of our conferences, as well as we also help our participants present posters from the project that is created. But of course, this is done in a long period of time because project takes a lot of time and depends on the topic that you're choosing. So that would completely be dependent on how much you have learned, how much knowledge you already have about the topic that you are going to do your project on. But besides that, this is exactly how it is done. And another question that I have is that how will we know which is a one-on-one mentorship, like who is our mentor for the one-on-one mentorship? So once we are through to the second week of the whole program, anyone who has an intermediate license, we will reach out to those people personally and ask them that if they have any requirement of setting up a one-on-one meeting with the mentor, or they can also reach out to us in case they have a question. And then it would be decided based upon that conversation, who would be mentoring them on that one-on-one meeting. So it could be a 1 hour one-on-one meeting to help them out with any of the course problems or any other problems that they would be having. So that is exactly how the intermediate session works. And the advanced license, as I mentioned, is again for not just the mentor sessions, but also the entire project session, and gives you another 30 days. So I hope that that is very, very clear to everybody. If you have any questions, again, you can email me, and thank you, Dr. Harpreet, for putting my email in the chat box. So you can go through all of these projects here. All of the participants who have actually done these projects, they have recorded this video. So you can hear from them how they've approached the project, what analysis methods they have taken. And if you have any questions, you can, of course, ask us. So again, if you have any question or query related to how to join the program, then please reach out to me at the address that Dr. Harpreet posted, marketing@pine.bio. And I also wanted to introduce you again to our groups in Facebook and in LinkedIn. So I just shared the posts on Facebook and LinkedIn. So you will be able to notice that we keep on sharing very informative articles, as well as talk more about such free webinars and upcoming programs in all of these groups. So if you're interested, please feel free to reach out to us. Okay, so Temitope has another question again. So what is the difference between the intermediate and the advanced license? So the intermediate license only gives you access to one-on-one sessions. You would notice that we have actually broken down the pricing. And while the advanced license not only gives you access to all of these basic and intermediate features, but also an additional 30 days, which means that your entire program access will end on June 20th instead of May 20th, which is when the program will end for anybody who has a basic license or an intermediate license. So this is completely up to you regarding what license you want. If you feel that you want an mentorship only, but you do not have that time to dedicate to an entire another month of working on a project, then you can choose this. Or if you just are a beginner and you're not very sure about how to do a project, but you're interested in just learning about what RNA-Seq is and how we can use it for understanding viral diseases, infectious diseases, then please go ahead and choose the basic license, because this will cover all of those resources that we have discussed. The only perk here is the one-on-one support, which is beyond a group session. So if someone is not very comfortable asking questions in a group, or wants some special time with the mentor just to express all of their queries, then they can choose for this session. So this is completely up to anybody. So if you already are part of the program and you want to upgrade, please write it to me via email so that I can help you out with that. So thank you all again for attending today's session. And if you again have any questions, please feel free to reach out to us. And I hope that you can also ask us any questions that you have beyond this session. And for all of our program participants, we'll see you again on Monday. I will send you all the email, all the details via email. And please make sure that you have created your account on Learn. That will give you access to the courses. You have your TBioInfo passwords ready. If you do not have those, please let me know.